Hi everyone, my name is Shivani and me here with my friend Shay are going to talk about influenza and what makes it good at evading immune system. So, do you know why do we need to take flu vaccine every year? Because unlike other vaccines like for rubella, mumps, HPV, Tdap, we just need two shots early in the lifetime and it gives you a long time protection. However, the flu vaccine needs to be updated and taken like every year. Why is that? The answer lies in the influenza virus itself. Influenza virus is built around 8 single-stranded RNA segments, which serves it its genetic blueprint. On the surface are spike proteins like hemagglutinin and neuraminidase. These spikes are not just for the show, but they are critical for the virus's ability to infect and spread. Also, they are critical for the immune response and vaccine to target them. So, here's when the things get tricky. They have various subtypes, and these subtypes come together to form different subvariants. This makes it impossible for the immune system to recognize or target them effectively. The evasion strategies are not just a theoretical concept. They have caused immense devastation. Think of 1918 Spanish flu, which was caused by H1N1 virus, led to millions of people's death. And many more, like 1957 Asian flu caused by H2N2, 1968 Hong Kong flu caused by H3N2, 2003 avian flu caused by H5N1, 2009 swine flu, which was caused by H1N1, and 2013 avian flu, which was caused by H7N9. Without effective vaccines, these events have led to widespread of illness, death, and overwhelmed healthcare systems globally. The influenza virus is a master of immune evasion, and it uses three major strategies. Antigenic drift, where small gradual mutations accumulate in the virus, and these changes are enough to make your immune system memory cells struggle to recognize the virus, even if you were vaccinated previously, because it leads to the formation of completely new variant. Antigenic shift. This is a game changer. The virus can swap entire gene segments with other strain and create a completely new subtype. These shifts are rare but have caused deadly pandemics like 1918 Spanish flu. Glycosylation, to make matter worse, when the wild type protein has diverse antibody response, whereas the virus, when cloaks itself by adding sugar molecules to its surface proteins, masks it from the immune cells and make it even harder to detect. This was about the virus. Let's try and understand how immune system reacts.
Hi, my name is Taiwo Luche, and I'll be speaking about the immune response to influenza virus infections. The immune response to influenza virus starts with the recognition of the virus by toll-like receptors in the infected cells. Typically, the infected cells are cells of the respiratory tract, so most of the infected cells are epithelial cells that line the respiratory tract. These cells secrete interferons when infected, and these interferons lead to a cascade of reactions subsequently. Interferons induce an antiviral response in neighboring cells. They cause apoptosis in other infected cells, and they also recruit white blood cells to simulate long-lasting immunity. The secretion of interferons by infected cells leads to the rapid recruitment of innate cells like natural killer cells to these infected cells. Natural killer cells are able to identify infected cells by the absence of inhibitory signals on these cells. This then leads to the secretion of granules such as perforins and granzyme into these cells, which causes cell death and apoptosis in these infected cells. Consequently, phagocytes such as macrophages and neutrophils arrive at this site of infections and then mop up these infected cells. Antibodies play key roles in humoral immunity. Antibodies bind to the virus, and then this inhibits the virus from being able to access the target sites on their target cells. Subsequently, they are able to inhibit the infection. Also, cytotoxic T cells are able to recognize infected cells by the presentation of viral antigens on MHC1 molecules. They then kill the cells by interactions of Fas and Fas cell ligand, as well as the secretion of perforin and granzymes into these cells. However, all of these reactions, all of this immune response, all are heavily dependent on the ability of the host cell and the host immune cells to recognize the virus. And this is heavily impeded by the antigenic drift and antigenic shift, which Shivani earlier mentioned. To solve this problem, researchers all over the world are trying to identify novel targets and novel sites on the virus which are more reliable compared to the hemagglutinin and neuraminidase surface antigens, which are prone to random mutations. One method which is being explored is the targeting of sites on the stem helix of the hemagglutinin molecule. This site has been shown to be conserved across different viral strains. So even in the presence of mutations of the hemagglutinin or different strains, the stem helix has been shown to have some heavily conserved epitopes which can be targeted for vaccine design. Other studies are currently looking into the development of vaccines which have as many epitopes, as many immunodominant epitopes which can be crammed into a single vaccine. These polyvalent vaccines are made from conserved epitope regions of strains of viruses, of the influenza virus.
Given the flu virus's ability to change and evade annual vaccination, it's essential to keep up with its evolution. The flu vaccine trains your immune system to target the most likely strains for the season, helping protect you and those around you. So stay informed, stay protected, and get your flu shot every year. Thank you.